Welcome to the Next Level Human podcast. As a human, you have a job to do. In fact, you have 4 jobs: to earn and manage money, to attain and maintain health and fitness, to build and sustain personal relationships, to find meaning and make a difference. None of these jobs are taught in school, and that is what this podcast is designed to do. To educate us all on living our most fulfilled lives through the mastery of these 4 jobs. I'm your host, Dr. Jade Teta, and I believe we are here living this life for 3 reasons and 3 reasons only: to learn, to teach, and to love. In this podcast, I will be learning, teaching, and loving right along with you. I'm grateful to have your company. Here's to our next level. All right, welcome everybody to the show. Today I have a really special guest for all of us. This is Dr. Mota, and she's a senior research associate at Cornell Medicine in New York City. She got her BS in genetics from UC Davis, has a master's in public health and infectious disease and epidemiology from the University of Melbourne, and a PhD in infection and immunity from Melbourne University. And she is the recent recipient of the Believe Young Investigator Award in 2019 for her work in HIV. And obviously, you know why I wanted to have her on the show. Dr. Mota, welcome. Thank you so much for taking time out of your busy day to hang with us. So, all right. So here's the thing. I just want to give everyone a little bit of background real quick. Obviously, we're going through this COVID crisis, and there's a lot of, I know you've probably been watching, all the stuff that's been kind of going on, and you're probably looking at it from a different perspective because this is what you do day in and day out. And so the first kind of question I want to ask you, just to give everyone a perspective on the kind of, background that you have and how much it takes to get a PhD in this area. Give us an idea of how long it took you and sort of your trajectory in virology and immunology, and how long have you been studying this stuff?
I think officially it was 13 years of tertiary education, including my undergraduate, my master's, and the PhD. And it's just a huge, massive accumulation of knowledge. So everything I learned since my undergraduate degree was in molecular genetics has contributed to the understanding of HIV cure science that I research now. And I haven't actually formally studied virology and immunology during my PhD. The concept in Melbourne is a lot different than PhDs in the United States. So there's no actual coursework or lab rotations. You're just thrown into the lab. And all of my previous experience, although I'd worked with HIV in the past, is mostly from molecular biology. So the angle of DNA, RNA, and protein, and the accumulation of knowledge through my PhD, just basically in reading the scientific literature. Every journal I read is a new piece of information to the puzzle of what to me will be an HIV cure. So yeah, imagine 13 years and tens of thousands of research papers accumulated here. Don't remember all of them, just the important ones.
I know it's a huge amount of, education that you get, right? And the reason I wanted you to just discuss that for us is because it's really interesting when you have someone who does some cursory, sort of Google research on COVID and becomes an expert in a virus, right? And it's really interesting. We were just talking before we hit record, and I was telling you, obviously I have a medical background, but this is a lot of this is Greek to me. We have our specialties, right? And so this is why we need to talk to someone like you who eats, lives, and breathes this stuff day in and day out. And so go ahead.
I was just going to say, when you say that, I guess a perfect example is if you and I were sitting in a park, maybe not Central Park in Manhattan, but say a park in Botswana, sipping on a piece or on a glass of wine, and say a mosquito came and bit both of us. Most people would be like, oh, that's annoying. I'm going to have an itchy little bite. But here I am looking for the little black and white stripes, which is, meaning it's the Aedes aegypti mosquito and a huge vector for transmission of disease. I would immediately be thinking about the plasmodium type of malaria that the mosquito could be carrying, or what types of viruses it could have. Are there flaviviruses, including Japanese encephalitis, dengue fever? Have I had dengue before? Is this a different endemic subtype? That means I could get hemorrhagic fever. And then, or is it Zika virus? If my sister's pregnant, should I stay away from her because the risk of microencephaly? So yeah, to most people, it would just be like, okay, I've got an itchy bite. For me, it's like literally thinking on the molecular level of all the pathogens that one mosquito may, but most likely may not be carrying.
It's a huge amount of understanding, right, that you're able to download, which is why it's so important for us to talk to you. So we're going to get into the COVID thing, but why don't you, if possible, give us, I mean, I know it's a huge topic, but help us understand just a little bit about the immune system. If you were going to try to explain it in a simple way for the average sort of American or average, person who's interested in thinking, hey, what is it about my immune system I need to be aware of?
Okay, so the immune system is the most phenomenal thing on this entire planet. It's a beast of a system. You can ask if I'm an immunologist today, I'll tell you no. 20 years later, I'll still probably tell you no. There's so much to learn. So if I were to break it down into two components, it would be the innate and adaptive immune system. So those are two different pieces of the immune system that are important to every single puzzle and are there fighting for you every single day, all day long, even if you don't know it. So the innate immune system essentially is that first line of defense after a pathogen breaches the barrier, such as the skin. So it's not specific to a pathogen, whether it's bacteria, parasite, dust even, or viruses. Say a pathogen enters your system, the innate immune system will respond within minutes to hours. It'll recognize something's wrong because these pathogens, for example, will have different things that are not pieces of your body, right? So there might be double-stranded RNA inside the cell, or there might be different ATP inside the cellular space, and your innate immune system cells will recognize something's wrong. So these innate cells are mostly phagocytes, so like macrophages, dendritic cells, and granulocytes. And essentially, they will come and try and chomp up and eat all of the pathogens away, or infected cells, or trigger the death of infected cells in a way that doesn't matter what the pathogen is, it will just do it immediately. So the adaptive immune system is where you develop immunological memory, which is a stronger, more specific. It's exquisitely specific, and it's not an easy thing to achieve, but it is achieved against most pathogens. So essentially, now you have your killer cells of your immune system, like your CD8 T cells. So I work a lot with CD8 T cells. So this cell will have one receptor called a TCR, or the T cell receptor. And essentially, before it can recognize something, it just has a sequence on it. It's just going around the body forever. And essentially, if you get a pathogen inside your lymph nodes, you feel your lymph nodes get all like clogged up when you feel sick. And that's because your CD8 T cells are going through there and essentially looking for the antigen that its sequence matches to. So as there's, for example, a virus in your system, different cells like the dendritic cells will be chopping up those pieces and presenting them and saying, hey, CD8 T cell, where are you that recognizes this? Do you know this? Do you know this? So many CD8 T cells will go by and not see it, not know it because it doesn't have the exact sequence match. When it finds it, that CD8 T cell will start to proliferate in a huge way and expand. And now that CD8 T cell is armed to find that specific piece of the virus. So now it will go around to the different tissues and start looking for it. And when it finds it, it triggers a very specific type of killing to neutralize the virus in that cell, to kill that cell. So that's the adaptive, the huge part of the adaptive immune system is the CD8 T cell, very, very specific killing based on very rare interaction of these sequences finding each other, versus the innate immune system, which will just kill things it knows aren't supposed to be there, even cancer cells. And the other side of the innate immune system also is the development of antibodies. So you have your T cells, which are the killer cells, and then you have your B cells, which in a similar way start to gain memory and then start producing antibodies that are against that pathogen, again, in a very specified way. So it's a way for your body to have huge checks and balances, regulations, and allow both the antibodies and the CD8 T cells to recognize pieces of the virus in a way that will be killing it specifically and not just killing every cell in your body.
Hmm. All right, so I'll give you a real world example to see if I can understand this, and you kind of correct me, and hopefully it'll help people understand. So if you and I are hanging out in a party in New York City, and this weird looking guy sort of walks in the room, and it's a cocktail party. He comes in with this weird costume on. Our innate immune system, the one that's sort of, looking for just general pathogens, is going to see this guy and go, something's wrong with this guy. And so if this guy pulls out a gun or something, the innate immune system is immediately going to jump on him because they were like, he just didn't look right. Right.
Yep.
So this is sort of like this innate sort of just globally looking for things that don't look right and then sort of attacking them. But then you have this other side of the immune system, this adaptive side that communicates, I guess, with the innate immune system to a degree to remember this type of person, that kind of shady activity, or sort of what this person may have looked like, or what kind of things that he may have been sort of wearing. And then we develop memory to this in a sense, so that when we see that again, we can respond faster. And maybe the guy who's, the bouncer at the front of the party doesn't even let this guy in. He gets caught really quickly before he can sort of do any damage. So is this, I know this is super simple, but is this sort of how the immune system works?
Yeah, exactly.
Okay.
And of course, viruses have ways to combat both the innate and adaptive immune system. And that's one of the reasons they're still able to continue to propagate and replicate, even though the immune system's trying to attack it at the same time.
So maybe a virus coming into this party is really adept at looking like everybody else in the party. So it makes it very difficult for us to sort of recognize.
HIV does that very well. On its spike proteins, called the envelope proteins around the outside of the virus, it also puts glycan shields or glycosides that have essentially sugars that look like the host, so that it's like shielded by what something looks like a host cell. So if the immune system's surveying and is like, oh, wait, no, this is us. This is part of us. Don't kill that. So that's one of the ways, for example, HIV can evade the immune system.
So that's interesting. So this is why viruses are so such a destructive force for our body, right? It seems like they almost are like have a cloaking device, whereas maybe bacterias and parasites aren't able to cloak themselves as well, and they can remain invisible to our immune system to some degree, which makes them really, really difficult to deal with. So I know you specialize in HIV, and that's sort of your major specialty, but let's talk about this virus, COVID. What makes this thing so different maybe? And also, I mean, one of the things I'm most interested in is they call it a novel virus. Help us understand what that means. Does that mean that all of us are eventually going to get this? Like none of us are escaping this? Is it a matter of we're all going to get infected? We just have to be able to fight it off, or will some of us not be able to get infected?
Yeah, so coronaviruses have been around for 10,000 years or so. And when they were discovered, there are different subgroups or subtypes of coronaviruses. And the ones that infect humans generally give us the common cold. And, you have a few sniffles, it's nothing to worry about. And of course, we've heard of the coronaviruses, SARS and MERS, which came or arose out of zoonotic transmission events from bats, and MERS came from camels. And essentially, this is a novel coronavirus because the sequence is different enough from previous coronaviruses that it has never infected a human population before. And therefore, no one has any sort of immunity against it. Just we essentially have to start fighting from scratch. So the only hope that we essentially have is a vaccine. I think vaccines are the single most important human invention, kind of like not letting that guy into the party. That's what a vaccine does. So while we have potentially immunity against other coronaviruses that have caused the common cold, we wouldn't have SARS immunity or MERS unless you were infected. It didn't really make it over to New York, for example. But the point would be that it's a new virus. Our immune system has never seen it before, and it's not as deadly as SARS or MERS, which was 10% and 36% mortality, but it's a lot more transmissible. And this virus is really unique because there aren't really any viruses out there where you can have 100% asymptomatic carriers. Usually, if you get a virus, you're going to have something. You're going to know you're sick. You'll know that there are some symptoms there. So the fact that this virus can go unchecked through a human population with zero symptoms and all the way to acute respiratory failure and death essentially makes it a very unique virus as well.
Yeah, this is the part that I'm so glad you brought this up, because this is the part that, not having your background in this, I was looking at this the same way. I'm like, have we seen a virus that does this? It's like this thing. One of the things, and correct me if I'm wrong here, but I think one of the things that we want is, sometimes we want a virus that kills very, very quickly because it'll snuff out really fast, right? So one of the things is, we hate that people die, but at least if people get really sick and the virus, does its thing very quickly, it can't spread real fast because people get sick, they're quarantined, they stay home, they stay in bed, they can't go anywhere. But with this thing, it's in us, we're shedding viral particles, and we feel just fine. And it sounds like what you're saying is, this is very unique for most viruses. And this is probably one of the reasons why this became a pandemic and went global. And also one of the reasons why perhaps it's been around much longer than we thought. We keep, like, as I look and try to research this, they're like, oh, it was here longer than we thought. It was here months before, we thought. At first we thought it was here in January in the United States. Now we're thinking as far back as October, maybe. So this seems like this is a big deal and something that you're sort of concerned about, right? Do we know about what percent of people who get infected are, I've read different things, 25%, 45% may be asymptomatic carriers? Like, do we have a number on that? Do we know? Probably not, right?
I have no idea, because it would be also the asymptomatic carriers that aren't seeking to get tested because they're not sick.
Yeah. And it's also one of the reasons why we have all these sort of changing, I think, and I guess this is a question, but it seems like this might be one of the reasons why we keep getting these changing sort of directives from the CDC and WHO, because it was evolving fast, and we didn't know that there were these many carriers. We also didn't know that we had these, people who can infect, these super spreaders who can infect many, many people. And so it's a really interesting sort of thing. Let me ask you this, because this is so interesting to me, and I've always wondered about this. How do you know how they actually find these viruses? Like, I've heard stories about viral hunters, like people who go to places and, get rats and study bats and anesthetize them and draw their blood and look for viruses. So I'm wondering, how much do you know about this, and how much can you tell us about how we actually isolate a virus like this, and how do we know this is an actual virus? Do we actually get the actual virus and put it in a petri dish and say, oh, there you are? Or are we looking for certain fragments of a virus? Like, how do we even know this is a thing? And this kind of gets into the whole conspiracy thing too, right? Where people are like, well, have we ever even isolated this thing? So I'm just curious about that science, how it's done.
All right. So a friend of mine during my PhD actually had been a virus hunter during her PhD for a hantavirus, which is a virus that caused the Four Corners disease, I don't know, a couple decades ago, and essentially was so airborne that if you swept it up in rat feces from your house, for example, you could be dead within 7 days, and you get really sick really fast and you just die. So she was looking at hantaviruses in Germany. And her virus hunting is that she puts on a full hazmat suit and goes for a hike in the forest and does exactly that, looks for rodents and collects their feces. And, if you're in Germany, it's a beautiful forest, and you come across a few hikers and they're looking at you like, ooh, should I be here?
Yeah, what the hell's going on?
What is that? But yeah, so essentially that is what happens, and it's necessary because we probably completely underappreciate the amount of viruses that exist out there that are already transmissible to humans. We just haven't been exposed to them yet. And I think a really cool example, well, it's not cool, viruses are cool, but they also cause a lot of devastation. An example of that is the Nipah virus, where essentially in Malaysia, they spread farmland for their pigs. So essentially they chopped into forests way deeper than they ever had before. And what they didn't know now was that the pigs, happily eating along, happily having their new farm space, were now exposed to a population of bats that have really never left their little bat colony and never really been exposed to other species. So they had Nipah virus. The bats and the pigs interacted in a way that the pigs got Nipah virus from the bats, and then the humans got Nipah virus from their pigs, and that sort of spread around a bit, and it was also quite deadly. So to have virus hunters, I mean, you have to look on every corner of the planet and you never know how it's going to mutate. So you might have viruses that are circulating in bats, something like Ebola or Ebola like, that never makes it to the human population, or that even if it did, would never jump species to humans. So it has to have, the receptor to bind into human cells and the survival in whatever our mucosal barriers are for respiratory diseases. But yeah, having virus hunters is really important, just to understand what's out there ecologically and kind of anticipate what might be coming. Kind of like we have to get a flu shot every year, because the antibodies aren't going to last forever. Also, you don't know what strains of influenza will be circulating that year, and it's all predictive of what's coming, how we get vaccinated for that. And what was the second part of your question?
The other part is, and this may be getting too much into science, this is just an interest of mine, but so like, what are we actually isolating? So when they get the blood of this rat, what are they actually isolating? Are they finding like a full-on, sort of virus, or are they grabbing seeing certain RNA sequences where they go, that does not look human, and let's go back and look at this RNA sequence and see if we have sort of a match? Like, I'm curious about these first infected people with coronavirus. How did they actually go, hold on, how do we know this is a coronavirus, and how do we know it's a new coronavirus, right? Like, what are they actually isolating?
So there are two parts to that, and technology is the answer to most of that, because 50 years ago, we wouldn't have been able to jump on this pandemic like we did today, kind of like 1918 influenza. Just the technology wasn't available yet to really figure out what it was fast enough to try and combat it. So one part of it is seeing the virus for real, is scanning electron microscopy. And I was looking the other day, and as early as February, we had electron microscopy images of the coronavirus. So this microscope is the most extreme detail you can see on any microorganism or small molecular level. It's essentially shooting a beam of electrons down at what you're looking at, and it interacts with the atoms on what you're imaging, and that creates a very, very structured, detailed image of what you're looking at. So in the early patient samples they took, and they used scanning electron microscopy to try and find a virus. And of course, morphologically, cells look a certain way in your body, tissues look a certain way, and viruses we have very well defined. We know what viruses look like. So very early on, they were able to produce images of the coronavirus, and essentially in that moment could identify it as a coronavirus because of the way the spikes looked on the outside of the virus. You could throw 7 different viruses at me, or images or drawings, and I could probably tell you that one's influenza because it has hemagglutinin neuraminidase. That one's HIV because it has GP120, GP41 trimers. That one's a coronavirus because it looks like a crown, and so on and so forth. So yes, we have isolated the virus, not me personally, but as a world, the virus has been isolated, identified, used in animal models, so already used in infection studies, so we know it's there. And then you talk about measuring pieces. So we can test the RNA. Sampling RNA from plasma from humans is a lot more simple than looking under microscopy. So, and we can do it a lot, batch it, do a lot more at once. And essentially, the way we knew it was a coronavirus and then it was a new one is, again, back to technology, is over the last 2 decades, ever since genome sequencing became available, there are massive, massive online databases of everything that's ever published in science. So it's called BLAST. You can sequence something and blast your sequence against everything that's known, and it'll be like, oh, it maps to this gene in this human part of chromosome, blah, blah, blah. Or it's HIV, it maps to that HIV RNA sequence. So when you had a coronavirus, and even if you sequence a whole cell or a whole group of cells, and there's so much sequence, lots of it will be human RNA, lots of it will be viral RNA. Your human RNA will map right to exactly where it matches to. The viral RNA will map to coronavirus in these databases, but it'll be like, oh, except for this region here is so different from what we've seen in the past. So because of this sequence technologies and the information upload and the worldwide collaboration of sharing data this way that's accessible to people who are scientists, we could immediately see the virus with the electron microscopy. The whole world could see it that same day. We could identify that it was a coronavirus morphologically and sequence-wise And we could also identify immediately that it was something new and we should be a little bit worried, or actually not a little, sorry, a lot worried.
Mm. Yeah, that's amazing, because one of the things you see for people who don't really understand what's going on is you hear a lot about, some of the conspiracy stuff where they're like, we've never actually isolated a virus. You're saying we have. Number two, we have actually sequenced this virus and compared it with existing sequences that we have to be able to say, here's the virus. We can see it's a coronavirus because of the way it looks under this, sort of electron microscope. It has this crown effect. So you being an expert in this, you can pinpoint and say that's a coronavirus. Then you got to go, well, is it a known one? And how likely is it to be something that we know? You sequence the RNA, you blast it out to this database, it sounds like, and then you say, okay, well, it maps to this particular type of coronavirus to a degree, but not completely. So now we know it's brand new. It must have, mutated, or somehow we've never seen this particular virus before. And then we can actually take that virus that we see, it sounds like, and I'm just repeating what I think I heard, inject it into animals and actually reproduce the COVID sort of infection. And we basically have these three lines of sort of corroboration now. We've seen it. We've got the RNA. We've actually then done infection studies on it and reproduced the infection in animals. And it seems like we've been able to do that with this particular virus. Is that what you're saying?
And then isolate the antibodies that are created against it and show that it binds to the specific sequence.
So it's really interesting once you get into the science of this to essentially say, listen, we have all this information here. It's not really made up. And I know this brings me to a side question, right? Where people essentially, when they look at science, you being a scientist and me having some science background, the way I've always seen science is that science is not making things up, right? And science is not, you're not using science to say, if Dr. Mota has a bias about a particular virus or whatever, she has to prove it with the science. The science is going to prove to her what the thing is versus the other way around. And a lot of people, I think, get upset about science because it's kind of this up and down, sideways process that it merges slowly over time. So do you have anything to kind of like that you would say to people who are listening who've never really been involved with science like you have about how we should be viewing this and our level of patience and why we should not be jumping to conclusions? I'm just curious if you have any thoughts on that or if it bothers you at all.
Yeah, so science obviously is not perfect. It is beautiful though. And you'll have people, so there are different parts of science. So the individual's mind, I think, is a huge part of the science they create and the discoveries they make. Because my mind, for example, works in such a molecular way that I'm thinking always about transcriptional initiation, RNA elongation, and splicing of HIV RNA, whereas the majority of my lab are already immunologists, so they think more about how the immune system interacts with the infected cell. So I'm thinking about everything happening inside that cell because of HIV, and they're thinking from this more bigger picture point of view. So first of all, we all have our different expertise, and we're defining my niche for my career. And essentially, there's so much scientific literature. So like I said, I've probably read tens of thousands of papers. I just did a quick search right before. So we have PubMed, which is the essential science Google library, and it's all the scientific manuscripts in all different sorts of journals. So this morning, there were 36,600 publications that related to the word COVID. So you can imagine, even if you were an expert in coronaviruses, since essentially February, who can read 36,000 scientific publications? They're not easy to read either.
Yeah.
They come with new information, new acronyms, new concepts. So it's really navigating your way through the information that's already existing, seeing, where there's holes in it, or not holes, but rooms for discovery, I say, holes for discovery. And then you think of an idea, well, this is what I know from everything I've accumulated in my mind and these studies and all the incredible, centuries of science before us. And then you come up with a question and you kind of form a hypothesis around this. Like, I think one thing's going to happen if I do this. And then you design your experiments around it. And then there's a lot of optimization. So you have to make sure your new assays and experimental procedures are working perfectly. And then you take time to repeat. And in different contexts, in vitro and cell lines in the lab, from cells from humans, inside the human itself, and in vivo studies, or in monkey or mouse models or ferrets for in respiratory viruses. And you just repeat until you've convinced yourself you've made the discovery. Then once you've done that, you write up a scientific manuscript, you put it out there in the world of publication, and 100 times the editor will reject it, say it's not good enough. But, you know, it eventually gets through somewhere. And essentially, then you have peer review. So they give your journal to experts in the field, and they all assess it independently, and again, based on their mind and their knowledge. And they might find things that they think you should have done, and then you can go back and repeat those and do those and add it to your study, or it might be perfect as is. Sometimes that happens, and then it's just set off for publication. So that's generally, of course, it's taking a lot of time to come up with all of this information, but we're in a pandemic. So this is pandemic medicine and pandemic science. So I personally feel like a lot of the information has been rushed, and maybe you've heard in the news that some big papers have been retracted. That means something was published, it was peer reviewed and published, and then it was taken away. That's very rare in the career of a scientist, but I think in a pandemic, you know, there's so much new information coming out every single day. It was just a respiratory virus. Now it seems like it's infecting or affecting the vascular system, the kidneys, the heart. What's COVID toe? I just heard that the other day. That's not your lungs. So I think that science usually takes longer than something quick, but the discovery of the virus, the sequences, and the way we can combat it is more quick because, again, I said all the collaboration worldwide of data sharing, and I'm working on this together. So the best sort of therapies that would work against it and the vaccine trials, that's like the fastest from the discovery of a virus to first in human vaccine trial that has ever happened in the world. And again, that's just because we do have so much technology and information sharing.
Yeah, and probably too, you know, we have a pandemic and people are sort of dying. So if I'm listening to you correctly, it's this science is this really interesting process where it's basically like the actual scientist, if they are basically honoring the scientific process, has to prove their hypothesis to themselves. And oftentimes they may prove that hypothesis wrong, and they do tons and tons of studies themselves, and then they will publish, and then that's looked at and sort of rejected, probably because these editors are essentially saying, we don't like the way you did this, or perhaps we don't see that the method matches, or whatever, or it's not good enough for the publication. And then once it's out and it does be published, then all these other people with all these various sort of lines of research that may be peripherally related come in, read your work, and then critique it and everything else, and then you kind of go back to the drawing board. And so you have all this that's just for one study. And then of course it looks like you were kind of educating us about all these different other lines of research, like maybe, and this is a question to you, like when I'm looking at this, not being a researcher like you, but being someone whose nose is in PubMed all the time trying to figure this stuff out, what I usually try to do is I go, okay, let me see what's going on, like in a pandemic like this. I say, let me look at the epidemiology, let me look at the population studies and see what countries are doing well, what countries aren't doing well. Let me see if I could tease apart, and we've looked at what they're doing that may be helping them or hurting them. Let's look at the actual viral studies and see what's going on there in animals and in humans. Like, for example, the mask issue, which is something that I think we know is a big controversy now, right? At first, when I was first looking at this, I was like, reading things on influenza and things like that I was like, well, we don't really know that masks work, necessarily. And so I can kind of understand why, some of these CDCs and WHO and things like that weren't necessarily mandating them. But then we start looking at new studies coming out saying, and looking at, for example, the population studies and saying, maybe they are, sort of doing something, to help these people, sort of stop this particular infection. And then we go and look at, the actual studies on masks and trying to figure this out. And so I guess the question there is, with you, how do we figure this kind of stuff out? So like with the mask issue, is this something where you're just like, oh my God, Jade, like honestly, like masks work and they've been shown to work? Or is this something like you're like, well, I get it. It is a little bit up in the air depending on the type of virus you're studying versus, the type of mask you're using versus all these things. So I'm just wondering if you have a comment and you can help us on that, because it's a big controversy right now, and I don't even know that it should be for someone who's a researcher like you.
Definitely wear a mask. I'll start with that. I think the type of mask is important. The context is important. A surgical mask, of course, and an N95 are the most protective of droplets not coming out and droplets not coming in. But I'd been wearing a mask since February, so everyone was staring at me like a weirdo, and I did not care.
So as a scientist, you're like, I'm putting on a mask because I've been working in this world for a long time. So you knew.
Exactly. Yeah. And essentially, the cloth masks are a bit more worrisome for me based on their permeability. So if you have like a really hot and humid day, and New York City is always very hot and humid, and you like notice a buildup of moisture in the actual mask itself, that's going to be more permeable for the virus to enter. So I think surgical masks at least should have been recommended since day one, but I also believe that we didn't have enough in the United States. So making that as a recommendation would have also been fraught. But also earlier on, they really thought this was a droplet virus, meaning I know it's hard to think about droplet versus airborne for the layperson versus the virologist, but to me, a droplet virus is you sneeze snot, it landed somewhere. The virus only is inside that snot. Whereas airborne, it's like you can breathe the air and it's coming in, like hantavirus from the rat feces dust, right? So that's just flying in your face and you're getting infected. I think the jury's still not out exactly. It's probably a bit of both. So it's in the droplets, but how tiny the droplets are. Now they say this invisible mist, especially indoors, can last 3 hours stagnantly, and you can walk through that and get infected. So there's that part too. So I think having a mask on, especially indoors, is essential. I mean, we're not allowed into our work building without a surgical mask on. And the other part of that is outdoors versus indoors does also make a big difference. If it's a windy day, the droplets even in the mist are going to be dispersed enough. And if you were to walk through it, there's a chance the tighter you inhale isn't strong enough to infect you. But I do think that everyone should have a mask, and a mask that's not going to become too much moisture laden. And I mean, until we have a vaccine, and we need between 60, but more like 80% herd immunity for this virus to go away. And just over 1% of the United States has been infected. We need 80% to have protective immunity, or this virus is not going away.
Yeah, and 1%'s been infected, and we have 150,000 deaths. So if 80% get infected, think about the actual numbers there, right? That's
and the death rate will increase. So the mortality rate of a virus is one thing, but the mortality rate is completely dependent on the structure of your healthcare system. So if you have no hospital, your mortality rate is going to be higher because you can't even treat people. You can see the different regions of New York City when we were inundated, and lower socioeconomic status hospitals had higher death rates, like up to 12%, whereas the really good private hospitals had maybe 1%. So that's also the type of patients they're receiving. That's also the type of care they can provide. You know, they were loading up dead bodies in refrigerated trucks outside Elmhurst Hospital. People were dying in the waiting room. Yeah. So that's just that speaks to mortality because of humanity, the way the healthcare system is. So the other part of the mortality issue is, of course, the way humans are deciding to adhere to these lockdown policies, wearing masks, being careful. And I know some states or some cities are doing better than others. I'm actually quite impressed with New York City. We've had, 23,000 deaths, and every day it was just rising. And, we listened to Governor Cuomo. He was an angel voice for us during this. And we have essentially flattened our curve really well. I'm terrified it's going to come back until there's a vaccine. It could come back any day. It just takes, one person to recede. But now that everyone's, aware and taking precautions, that one person could recede 80, like how it started from the person who flew from Miami.
Right.
It's more unlikely to happen because we all know now, and we're all doing things to prevent it. So yes, just wear a mask. Make sure it's a good enough mask.
And, it sounds like you're also saying, hey, listen, make sure it doesn't get too moist, clean it, that kind of thing. If you can have a surgical mask, do it. If you can get an N95 mask, even better, although leave those for the professionals who need them. I know I want to really talk about vaccines because that's another controversial thing. But before I do that, you mentioned sort of different strategies, and Sweden is a country that everyone's talking about. Obviously, it's got a population of about 10 million. So that's a very small population. I don't know that you can, possibly say Sweden is going to be like the United States. I mean, how many people are in New York City? Probably more than probably more than the whole country of Sweden, right? But at the same time, they decided, maybe because they have such a small population, to do the herd immunity strategy. And I don't know how much you paid attention to this, because some people are saying it worked, some people saying it didn't. I know they have, I think last time I looked, we had a fatality ratio of 3.3. Theirs is double ours. Okay, yeah, so theirs is like 7. And so some people are saying it worked for them. Some people are saying it didn't work for them. How much can we look at some? And I know one of the things that I loved about you is that, you have a master's in public health. And so you're very familiar with epidemiology. Yeah, right. So, epidemiology is a specialty of yours. And so you are incredibly qualified to speak on this idea of like what they're doing. Are they doing it right? And does it make sense for different countries to have different strategies based on population? And should we use Sweden as a model, or can we even do that? I'm just curious if you have thoughts on that.
So actually, my master's was done during the H1N1 2009 pandemic. So we studied it very intensely as it was happening. But I think it's the wrong question to ask if it worked or not, because I think it was just so irresponsible to begin with. We have a novel coronaviruses. When coronaviruses are bad in the human population, historically, they're really bad. So we didn't know anything about it. We didn't know what it would do in the human population, what direction it would go in, if it would mutate and become more pathogenic and have a higher mortality rate. We didn't know the immunity that it would create. So, I mean, just like dengue fever, if you have subtype A dengue once, if you've had it, it's called bone-breaking disease because it's very painful. But if you get subtype A again, you're immune for life. Your immune system's like, oh, I got this. Let's get rid of it. But if you got a different subtype of dengue fever, your immune system's like, oh, I got this. Let's get rid of it. And then it's like, wait a second, it's a little bit different. I'm not sure. And it just overstimulates itself and creates hemorrhagic fever. And you are more likely to die if you get a second subtype of dengue. So we don't know if the immunity against a coronavirus would have any sort of weird immune interactions with the coronaviruses we've been exposed to as a common cold in our whole history of living. So
you see it as taking a huge risk that Sweden took a big risk.
Yeah, and with the herd immunity thing, that assumes that you were even going to establish immunity. And now we know lots of people don't develop antibodies. Even people who've had severe COVID don't develop antibodies. Even people with antibodies in the lab, we're seeing they might not be neutralizing, so they might not kill the virus. Like most people with HIV, they don't make neutralizing antibodies against the virus. And we're seeing they're lasting about 3 months. So if you've just put your whole population through that and then lost people because of it, and then in 3 months, it doesn't matter because the antibodies have gone from your body. Not all of them were going to protect anyway. And anyway, not everyone got the antibodies. So I thought it was a bit irresponsible with a novel, terrifying virus that we knew hardly anything about to just say, no, it's going to be fine. We'll just do it this way. So I'm glad that New York City didn't do that. We would have all been dead by now.
Well, so let's talk a little bit about this idea then of like, because you specialize in HIV and there's no HIV virus, correct? I mean, you're essentially using antiretroviral drugs to deal with that. Am I correct in saying that?
In plasma.
Okay.
Yeah, so HIV is a different type of virus. It's a retrovirus, so it means that its genetic material gets integrated into our host DNA. So once it's there forever. And it's resting. We call this viral latency, where the virus is essentially hiding or sleeping. It's not actively replicating because, the antiretroviral therapy is so fantastic. It completely stops viral replication. And also, if someone's taking their medication every day and they don't have a detectable viral load, they can't transmit the virus to another human that's uninfected. So we call that U equals U, untransmittable, or undetectable equals untransmittable, which is such an important concept in reducing stigma as well. But yeah, so with HIV, the therapy is fantastic. We just can't get rid of that hiding infection. But if someone were to stop taking their medication, viral rebound usually happens within 2 weeks.
I see. And with something like this, then can we actually? I've heard people say, can we actually get a vaccine against this particular virus? Are we going to have to use drugs? Like, for example, even influenza is something that we don't really have a true vaccine for, right? If I'm correct, because the influenza changes and there's a new vaccine that has to come out every sort of year, and it has varying degrees of effectiveness. So what's your take on educating us with, will we have a vaccine? Will it be effective? And from your perspective, it sounds like you're sort of like, vaccines are the only real thing for, if I'm reading into sort of what that can really save us, if I'm reading your language correctly.
Yep. So vaccines are the single most important human invention, full stop. I don't think without vaccines and antibiotics, but mostly without vaccines, we might not have the world the way we have it today. So yeah, a vaccine essentially helps you to elicit that immune response. So if you're infected with the pathogen, then your body can fight it off and you won't get infected, just exposed. And that has saved millions and millions of lives, particularly in children in the developing world. And I mean, we haven't seen how it worked in our world because it's been so long since anyone's seen polio, smallpox, completely eradicated from the planet because of vaccination. So there are a lot of vaccines that are under development at the moment, lots of different types of vaccines as well. And the important thing is that they go through the safety trials, all the phases of the clinical trials rigorously. And this is important for a lot of reasons. Of course, just safety from being injected with something, sure, great. All of them have been safe that way so far. Second, you want to know if they have elicited an immune response. So did the person create antibodies because of the vaccine, and are those antibodies neutralizing the virus? So those are the two main things that you want to study in smaller groups. And then when you expand the clinical trials, you want to see if it works in a large majority of people and the type of immunity that is elicited that is actually protective from acquiring the virus. So, vaccine trials take years, generally speaking. This one is super rushed because we are amidst a pandemic. So usually you have the luxury of time. We don't right now. So at the same time of doing the phases, I mean, as a scientist, I would do one, then the next, then the next. But here we're doing them like one side by side, but we're getting the same information essentially. So even if they're being done side by side, because there are so many different categories of vaccines being trialed right now, one of them will work. One of them will be the leading candidate. And I know there's also been funding, for example, to start mass producing these in case that is the one, so they can be immediately deployed as soon as the scientific community gives the green light, and then the FDA gives the green light. So it might seem like it's going a bit fast, but I believe that all the precautions are being taken. And the most important part of the study is to do a huge trial and see that people are actually protected from acquiring the virus.
So you sound, if I'm hearing you correctly, you sound pretty confident that we are going to have a vaccine to this. Am I reading that correctly? That eventually we will have a vaccine and that will be the solution here, or are you? Do you have any sort of doubts at all?
I mean, I guess I had doubts previously because RNA viruses are typically harder to create vaccines for. I mean, HIV doesn't have one, but it's a retrovirus. Influenza is changing all the time, and we do need to get revaccinated each year. But some of the preliminary data suggests that this does create a protective immunity. So the only way to further test that is to wait and see what happens. So I'd say, I know I've heard recently the end of this year, but I'd still say, mid next year sounds about right. I mean, it's not also like you could do the clinical trial in New York City anymore because we flattened our curve so well. So do 10,000 vaccine trial people and they're not being exposed to the virus. So you can't actually test the.
Yeah, interesting.
Right. And so, yeah, you have to have, you have to have the virus around to be able to test the efficacy of the vaccine. I heard some podcast there, this doctor was describing they should do forced injections in a controlled manner, which is always a possibility, but I don't know anyone who would personally sign up for that one.
Right, right. I mean, there's ethical things there and everything. I guess one of the things that, well, I'll just ask you, would you, doing that, you do this medicine day in and day out, would you get a vaccine? Would that be something you would be
Oh,
confident in doing? Yeah.
100%.
Okay. And I guess the reason I asked that question is because certainly you have heard and out there in the world, right? Like you're in this world where you're studying. Certainly you've heard people saying vaccines are not safe, vaccines are going to do damage. There's all this controversy around vaccine safety. So I don't know if you have any comments on that for us, for people who are listening, who are like hearing all this stuff out there, but they don't get to talk to an actual immunologist, virologist. And so you being sort of the expert here, what's your take on that? And what would you tell people in terms of this information that they're getting about is there any, sort of safety concern with these vaccines in general? And what do we need to know?
So I'm always interested because you're right, I'm not exposed to anti-vaxxers on a daily basis. Um, I'm kind of curious as to what their main points are or what they think is not safe about a vaccine these days because, back when vaccines were first invented and there wasn't proper storage, yeah, a few children died of a bacterial infection, but that was, something based on hundreds of years ago that just wasn't really in the context of how today vaccines are manufactured and stored, for example. So
Yeah, I think the main thing, if I could summarize what I typically hear, it's basically that a lot of it started with young children going through sort of their vaccine regimen and then having things like autism and
right, okay.
vaccine-based reactions and all of this kind of stuff. Which, if you have something to say on that, because I think it's something that's where this started. And my understanding is that was. Well, I'll let you kind of go with this if you have anything to say.
Yes. Okay, so Mythbusters here now is that vaccines do not cause autism. And there was a study originally that showed that vaccines were associated with autism, possibly. This scientist, unfortunately, it was a bad seed. There are bad seeds in every industry, and his data were not real, and it was retracted. So that's one of those studies that was removed. And now, time and time again, through such larger sample sizes, have solidly proven that vaccines do not cause autism.
And I'll just throw something weird at you, and I just want to see what you say about this, Dr. Mota. So what do you say to the people who say, well, it's all one conspiracy. Everyone wants us to get vaccinated because they want to control us and they're hiding the data and all that kind of stuff. Do you feel like, what's your thought with that? Do you just see it as sort of like, oh my God, I don't understand what people are saying, or like, do you have any thoughts on that? Because it really is, the reason I'm pushing you on this just a little bit is because it's not every day we get to talk to someone with your expertise.
Yeah.
And so I'm just curious, for someone on the front lines, like, what do you even say to that? And
well, I guess the first question I'd ask them is, do they own a cell phone?
Mm-hmm.
If they're concerned about information and sharing information. So there is nothing in the component of a vaccine that inserts any sort of chip that records any sort of anything from your body. I guess I don't understand where these conspiracy theories come from, but a vaccine is there to protect you from deadly pathogens. It's there to protect your children. And I think we lack appreciation for vaccines because we are not amidst the death. So people used to have 10 children to hope 2 made it past 5 years old because the under 5 mortality rate from now vaccine preventable diseases is horrible. So many children died, and we don't see children dying today. I'm sure if it was still like have 10 children, so 2 survive. Oh, but if you get vaccines, you only have to have 2 children to have 2 survive. They will be way more likely to still believe in or see the actual benefit. I think not being able to see the benefit is one of the huge problems with people believing in vaccines, because they just don't understand the biology behind it, and they don't appreciate how many millions of children's lives have been saved
Yeah.
because of vaccines.
Yeah, it's almost like this thing, you don't know what you don't know. And if you're not, sort of in the research world and seeing this stuff, and you also didn't live back when people were dying of, smallpox and polio and seeing those things be eradicated, that perhaps even if there was, in my mind, and there, perhaps we'll find out there is some small issues of vaccines, some reactions that people have with vaccines. it it probably pales in comparison to the lives that have been saved, in my mind. I know for whatever reason people will listen to this podcast that I'll be controversial to them, but I'm just going strictly on the data, and certainly willing to, if that changes, to amend that. I guess we're running up on time, and I want to be cognizant and aware of your time. I'm so appreciate of it. But let me ask you, sort of, I'm a naturopathic physician. So for me, lifestyle medicine is everything. So I would say, okay, masks, sure. Social distancing, sure. Vaccines, sure. But then there's this other idea, and this is sort of the final thing I kind of want to get into you, versus sort of this idea of the sort of hygiene hypothesis and the terrain hypothesis, which essentially goes like this: that if we humans take good care of ourselves and are super healthy and eat all the right things and exercise and all that kind of stuff, then that is our best protection against viruses. That's the first part. And then the other part would be, if we overly clean our environment, we're not exposed to pathogens, we don't get sort of these bacterial samplings with our immune system and things like that, we're actually doing ourselves a disservice as well. And I'll give you sort of my take on this, and I just want to see yours. My take is that I think, obviously, if you're a super healthy human, that's going to help you perhaps fight these viruses better. But I'm not so sure it gives you complete immunity to things, because these things, our immune system doesn't necessarily work like that. If it hasn't seen something before and you get infected with that, whether you're healthy or not, you could still succumb to this disease. I'm interested there. And then I do have this concern about, well, I do feel like if we're overly cleaning everything and not exposing ourselves to anything, is that having a negative consequence on our immune system? So do you have any thoughts on what we can do personally to bolster our immune function, to become healthy individuals, to help perhaps with this disease and others, and also this idea of this hygiene hypothesis and the fact that we need our immune system to be competent and flexible and strong needs some exposure to pathogens? Any thoughts on that before we wrap up?
Yeah, so to the second point, the hygiene hypothesis, I completely agree with 100%. You need to let your children eat dirt. You need to have your children exposed to everything in the environment, because that does build your immunity and your memory immune system, or your immunological memory that can last for your lifetime. And particularly drinking the colostrum, so the first milk from your mother, that has all of those antibodies to protect the infant until the infant can start developing its own immunity. So all of those things are very important. And I agree that, I mean, I don't really have bleach products in my house or antibacterial soaps at all, because I also don't want to over sanitize anything in my house, or in my environment, because except for in the lab, where we're working with high titer viruses, there's a lot of bleach and UV there. But at home, and for mothers and families, I also agree that they should not be over sanitizing. And those small pathogens that you're exposed to on a daily basis, that your immune system is fighting something. Every time you put something in your mouth, every time you eat something, it's going to be laden with bacteria one way or another. And I think that the immune system's always working to fight, but it's when one crazy pathogenic organism comes inside you, and essentially it just is super deadly. It doesn't matter how strong your immune system is, and everyone's immune system is so diverse. So going to the first point, I think what's a problem, and I don't really understand what people say when they're saying they want to enhance their immune system or boost their immune system. So the perfect example is COVID. It's people who are having too strong of immune response, creating that cytokine storm in their lung alveoli, which is actually what's killing them. It's their immune system that's killing them at the end of the day. The virus is there and replicating in cells, but it was the immune response and all of this stuff, inflammation, everything coming to the site of infection, because it's like, oh, we got this, we're so strong. But it was the strength of the immune system in that context that actually is killing the person and causing them to essentially drown in their own lungs. So COVID is an example when you don't want the strongest immune system. There are interesting research articles out in the scientific literature that show different specific components of the immune system that people with immunodeficiencies might have actually protects them from viral pathogens. Again, because it's relying on the inflammation that really sustains viral replication. So to me, boosting an immune response makes it seem like you want more fighting cells, but the more fighting cells you have, there might be more of a chance you actually kill yourself, or they become autoimmune and attack your own organs, or just provide too much strong inflammation. So I also live by a very healthy diet and lifestyle. Well, maybe not during the pandemic as much, but
With all that good food in New York City, I'm just saying.
yeah, exactly. But yeah, so I mean, taking vitamins and antioxidants, those things are going to just help your system generally, and they're not bad for you, of course. And whatever your body doesn't want or need, it will end up excreting. So, being stuck inside, vitamin D anyway is always going to be helpful, beneficial to your body. But yeah, I think the only thing to really enhance your immune system in a way is vaccination.
Yeah, so maybe we should start talking about we want a refined and enhanced and sophisticated immune response versus this sort of robust, stimulating immune response. And it's interesting to hear your take on the sort of the terrain hypothesis. So essentially, you're essentially saying, yes, be healthy and all that kind of stuff, but we need both. You need to live a healthy life and do all those things, yes, but we're also going to probably need vaccines if we're really going to tackle this. And of course,
definitely need vaccines.
Definitely going to need vaccines. And then the hygiene hypothesis is something. So I guess the final thing I'll ask you, as someone who's on the front lines of this and doing, this work and understanding it, what are you personally doing, and anything else you'd want to share with us? Like I'm curious what your personal regime is. Are you doing anything like zinc and N-acetylcysteine and certain things to help yourself, or making sure you're reducing stress, or any other things that you feel like, Jade, we have to cover this to kind of help people understand? I'm just curious for you how you're personally sort of tackling all of this kind of stuff.
Well, my sister sends me a lot of awesome stuff, and I eat all of it. So I get a lot of different supplements from her, and powders like green juice powders, red juice powders, bone broth powders, and I put them in everything, and collagen hydrolysate powders. I think, just things that keep me generally healthy and fit. And I eat a lot of cheese. Sometimes have a wine. I think, whatever, and chocolate, whatever makes you happy is also an important.
Well, that's reducing stress. And there is a line of research, by the way, that the French paradox may be coming from blue cheese rather than wine because of some of the impact it has on the gut microbiome, just so people know. But so maybe that cheese is doing you some good. All right. Well, I just want to say, thank you so much. I mean, this has been amazing to talk to someone with your background and your expertise. It's been really difficult for me to find anybody because you guys are just all so sort of in the work and, not really available. So your time has just been amazing. Thank you for your generosity and the work you do. And I know you do a lot of other things that are volunteer work and everything else. You're just an amazing human, what I would call a next level human, and just so appreciative of you. Thank you, Dr. Mota, for your time.
Thank you so much.
Where can people find you if they want to? I don't know how much you're on the social medias and things like that, but if someone wanted to kind of follow your work and pay attention to you and just keep up with you, where would they find you?
So on Twitter, I think it's Talia Mota, so TALIA _ 1 or 2, I'm not sure, and Mota. I'm generally doing more of my science retweets and my own publications posted there. I have an Instagram that shows all the other fun stuff, and I think Talia Monique. Monique's my middle name. That's where I am.
Dr. Talia Mota, thank you so much for your time. You've been wonderful. Thank you.
Thank you very much.